Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Bin Yu, who's Chancellor's Distinguished Professor at UC Berkeley and a pioneer in interpretable machine learning. I came across Dr. Yu's work as I was scrolling Twitter, as you do, and saw this really interesting paper that questioned one of the things that is almost like a dogma in the field of linear models and genetics. And it was a really interesting piece of work out of Dr. Yu's lab, but also working with you and Ashley and a number of other co-authors that looked at epistasis in cardiovascular disease and cardiovascular development. I wanted to have Dr. Yu on the podcast today to talk a little bit about her work in general, but this concept in particular of nonlinear modeling and statistical genetics. So Dr. Yu, thank you for taking time to be with me.
It's my great pleasure to be here. Thank you for having me.
I would love to start with your perspective on linear models in genetics. Those of us who have advanced degrees in genetics, kind of have it drummed into you that you should first think of a linear model, and most GWAS statistics are additive, and we have a whole ton of work, and that obviously tends to be the case in general, but it maybe feels like we've tilted that direction to assume that it's always only the case, and you've done a lot of really interesting work looking at epistasis and nonlinear models in general. What's your perspective on what is behind this, and talk about a little bit of your work that's questioning that dogma a little bit?
So I think linear model definitely is bread and butter, right? When a teacher applies to machine learning, that's something you can really understand analytically, and there's just so much you can say. So it's definitely one of the, I think, bedrock for our profession. But I also think over the last 100 years since Fisher started on this path, that a lot has been understood, both from genetic point of view, also from statistical methodology, machine learning point of view, right? We really moved away, especially, pioneered by my colleague Leo Breiman, and many of us jumping in this interface between machine learning and statistics. We really went more for predictability, right? Without the necessary generative model. Really, that's something I really embraced for 25 years. It just really free you to directly go for this reality check. And you can go back through model distillation, and it's just a very new way of thinking about things and kind of remove a lot of the burdens of generative models. And sometimes maybe it's not the best, but a lot of times I felt like give you a foothold in first base, that at least you have predictability. And then you go for interpretability and mechanistic interpretation, right? So it's kind of the other way around, right? Classical statistics, maybe there was limited data. You have to use more conceptual way of coming up with linear models. But often if you look at the practice linear model, for my taste, we don't check the assumptions enough, like additivity. And we quickly go to calculating p-values. And that's concerning because if the probability theory is solid, but if you don't do enough checking, then all these p-values are not on solid ground. So that's like a self-critique, not just in genetic, in general, right? We haven't emphasized enough model checking at all. Linear model is a lot more assumption than doing a predictive model.
Yes.
And predictive model can be directly checked through not perfect test data validation. It's not perfect, but it's a lot better. It's much weak assumption than you assume additive model, Gaussianality, asymptotics, right? If you use p-values, we have weak convergence, actually. In my class, I say that if you really want to read off a p-value from a normal distribution, assume all assumptions are correct, you need to look at strong approximation with something like as a Berry bond, and that gives you 1 over square root of approximation error. And if you want the p-value 10 to the minus 8, you might roughly need at the sample size 10 to the power of 16. Otherwise, you cannot read out this approximation because asymptotics it should be seen as approximation, but the mathematical result we often prove is weak convergence. As the name says, it's weak.
Yes.
Therefore, I think there's this even mathematical gap using these p-values. And on top of that, you have model checking. So I think if you look at the pipeline, all the assumptions we put in for linear models, there are many, many weak points we're not really checking. Yeah. And therefore, that put questions into the final result of this pipeline.
Can you, to help people understand this a little more deeply, I'd love to ask you to talk through an example or two. And you had a paper in 2024 that is probably the simpler example, and then we can move to the more recent, more complex one. But do you think the epistatic interaction around red hair or MS or maybe both is a useful way to explain this concept to people? It would be great if you could talk a little bit about that finding.
Yeah, maybe I have to go back to a 2018 paper on predictive stable interactions, right? Which I shall mention my senior colleague, Ben Brown, right, who really got me back to genetics. I worked a bit in genetics 25 years ago and kind of went to neuroscience. And I tend to be a generalist and go for interesting problem. I have this deep belief that things are connected, things will be simple, and we can borrow like insight, wisdom from one problem to the other, right? So a lot of that is kind of, I also believe statistics is really a hub for ideas. Therefore, I cannot just develop a method to solve one problem. And I'm very much into this in-context research. I want my methodology to be developed while I solve a scientific problem. And then I generalize. Instead of I devise a method, always a hammer look for a nail. I just felt like my personal belief that the impact of such a method in context development has a lot higher chance to be useful in another real problem. And so there was like random forest at the point was already very useful in genetics, right? In predicting bioinformatics, different disease was one of the forerunners of tabular data. And then two young people, this is Basu at Cornell now, Kankumbir now a startup. And so we come together and thinking that, but you cannot interpret random forest. That's a lot of the kind of pros for linear models because you can interpret it, right? So I think interpretability I definitely care. But for me, is that the first thing is that have you reality checked the model before you want to interpret, right? For model development, be like deep learning, you might want to do that for training. That's a separate thing. But usually we try to interpret the final model. So one block of this random forest stuff, which is Leo Bryman really made very useful, and among others, is that people already had this idea that you have random forest and two features of genes sit on the same path of a tree in a random forest. They might interact, right? So of course, this is in two ways differ from deep learning model. It's not fitting additive model main effect first before you go to the interactions. It's directly go for this predictability, right, of this random forest, which we know works really well. And then you just look at this collection trees, and then you look at the two genes on the same path. The first one is to predict the enhancer status with Drosophila data with many of the genomic kind of typical features. And then the problem is that people had thought about that, but nothing was stable, right? You cannot, it's common sense. If things just come and go, you don't think that's knowledge. So we said the field was kind of not really progressing. So when we came to the scene with my collaborators, we thought we need stability. So at the point, I was already really had a paper called Stability, right? With like a Bernoulli Tukey lecture, which I was fairly in love with stability as a general concept, general principle. So, and Ben probably know that paper 2011, 2013, and came to me and with this like, we probably need stability here. And took good thinking and to realize that we need stability in the sense that we need to build in some soft dimensionality rejection. That's one way to build in stability is regularization, right? But the devil is in the details, right? You say, well, I want to regularize, but how do you do it? So the idea was that we start with random forest, and random forest already have this importance measure called MDI, right? And then we use that for the next iteration, we don't do uniform sampling of the features as in random forest when you do the split. We use weighted sampling, right? So we're already saying that some samples or some features are more important to MDI. You can replace it by better importance measures. It's very modular. And also thinking is that some genes are important, only appear when you see them interaction. If you look at individually, they don't come up, right? I think that's where the frontier for functional genomics, the GWAS, did pretty well for these single important genes, right? But the polymorphism is really about genes interacting to be important. So that's where I think that this random forest, we call iterative random forest, give us the ability to bypass, go for the top important features, like in linear model. We typically for important features when we look at polynomial interaction. So there are two things. We bypass this important genes for, of course, even for important sampling, it's really, really not very important. You're not going to see it either, right? So we kind of move from the top ones to medium, important, give them a chance to jump to be the important genes right away without saying, well, you're not important enough for the first cut, therefore we won't see you, won't study you.
So I just want to give a quick example and try to make sure that I'm tracking, the audience is tracking. So if we start with like a toy model of a random forest, you'd be saying, let's say we have 3 genes or 4 genes. You're looking at if we have this particular rare variant in this gene and that particular rare variant and not the other one, or like we have the rare variant version of A and B but not C, or we have A and B and C. You're looking at the collection of these trees that fit the data best, and you're saying as you run through iterations of the modeling, you're subsampling in terms of the next distribution in the trees that fit the data best, so that you can get a sense of what combination of features, besides just simply adding their presence or absence together, but conditioning on the presence of one or the other explains the data best. Is that a reasonable explanation so far?
Not exactly right. I think for rare genes, we have an ongoing project that you have to kind of do some translation into more representation, right, to do that. But so the rare genes we're dealing with right now, right, hopefully we'll have something in a couple months. But this is like, for example, go to the cardiology, right? This is hypertrophic cardiomyopathy, 1 in 500, right? So it's not too rare, but it's not too common either. So that's where we show the work. So you're right that we try to look at fewer genes, but the way to do it is through the importance measures of random forest. Suppose we have 3 genes, and the 3 genes got ranked into 1, 2, and 3, and with probability 0.5, 0.4, and 0.1. So for the next iteration, random forest usually do uniform sampling with some size, right? And what we do is we give the first gene 0.5 probability to be sampled, and the second one 0.4 probability to be sampled, and the last one 0.1. So the last one get a chance to be seen too. If you go for additive model, you might just don't want to look at it.
Yeah,
because it doesn't seem so important.
Okay, that makes total sense. And now I'm probably jumping to genetics a little bit, but one of the big challenges is we have so many loci in the genome that you can measure, even if you just focus on the common ones. Yeah. And you often have relatively few samples to work with. Yeah. So some way of constraining the combinations, right?
That's a great point. We actually do use GWAS in this very large feature space to prioritize. And we bring in stability again, right? So we take 2 GWAS to typical package and do a union. So we don't want one particular like plink or something dominate the next one. So we take 2 or 3, whatever, and we take a union there, aggregation. And then that's the pre-screening, and then we go to the iterative random forest step. So
I see. So you start with a smaller group of candidate. Yeah.
So we do use GWAS to screen, but we go to thousands of them, right? We don't go to like 5 of them or 6 of them. So you give, for the very, very, very non-important, nobody will get see it's probably not, right? So we do use GWAS to go to a few thousand, like 1,000 or 2,000. And then we go to this kind of non-linear model selection mode through iterative forest, right? And then we just have smart ways of collecting this overlap. And then we do another outer loop of bootstrap, and we get the stability score. That's our original paper called the iterative random forest. And later we have a sign version, because for genetics, it's important to know genes is overexpressed or underexpressed. So we can use that to call signed, because we say this gene is up or down. And that's the version we mostly use. And the paper is on archive. We'll actually put a lot of the enhanced results on the UC Santa Cruz browser for other people to use, but paper is not published because most of the ideas already in the original paper. But we did work very hard to have a track in the UC Santa Cruz browser for people to look at the epistate interactions. Yeah.
Interesting. So because maybe you did share hypertrophic or cardiac hypertrophy, I guess, as the phenotype. Can you talk a little bit about how you'd apply a model like this in cardiology to get a better understanding of the nonlinear interactions that are going on?
Yeah, this was a great opportunity that Chancellor BioHub have intercampus work. That's why you and Ashley and I work together with other collaborators. And it was pretty challenging because the first few cardiovascular phenotype we tried, actually, we didn't see any signal, right? And then we did do earlier work for red hair, right? So that was a simple genetic trait, and it worked with this iterative random forest. But we went to cardiac disease, the HCM. So we tried at least one or two other phenotypes. It didn't work. And then we went for HCM, which is expertise you and Ashley. And the label, we couldn't beat random toss. There's no signal for a few months. That was right before the pandemic. And then, as I mentioned, Tiffany Tan, who's in Notre Dame now, and then Chen Wang went by Jiao Tong University in China. So these young people, which really did a lot of the heavy lifting. And so we had to go back to the drawing table. And in hindsight, it's because I think many people have HCM, but not diagnosed. They were false negatives. And then, so at the time, deep learning already became a thing, right? So we use somebody else, like algorithm, to went to a smaller data set with people with MRI, right? And then we find the volume, as some of the audience probably know that HCM, the symptoms is really the left ventricle chamberal wall gets enlarged. So we got that volume. And then we also follow this PCS framework for vertical data science. P means predictability, C means computability, S means stability, right? It's expansion of statistical framework, and married with machine learning. So there we follow the framework. P means later extend to reality check. We now also have a book. So we said for continuous, we have to normalize with height and weight sensibly, of course. But we said normalize left ventricle mass. We still have the problem. How do we know we're not fitting noise, right? It's a regret problem. We don't know it's the noise level. So what we end up doing, I was doing a lot of other medical kind of work. For other products, we binarize things. It's as a kind of denoising. So I said, let's denoise it by taking the top, like this regression measure called LVM, and then the bottom normalized by height and weight properly, and then make it into a binary classification problem. We also make the signal clearer, right? Because we're cutting out people from the middle, right? We take top 10%, bottom 25%. We're forcing a clean signal because we're kind of doing AI for science. We're not really trying to find a treatment. We're really fighting for like recommendation for experiments. And that gives us a way to assess the P, which is reality check. If we cannot beat the random toss, we shouldn't continue. And we beat it very barely by 5%. We got 55 because it's really, really hard, as you know, I'm sure the audience knows, the complex human genetics. The good thing, the consistency stability is that if you cut at 15%, 25%, all of that stability, it holds. If we change the linear model versus random forest and iterative random forest, it holds. So there's this consistent signal against the stability, says that there's something there. And for interpretation, because we believe this, there are a lot of biological processes. It's interaction is actually tree-based, like you need abundance of biomolecule for them to interact as, I think, a really serious alternative to this polynomial interaction. And we use that iterandom forest or sign iterandom forest as interpretation model. And there we have to add more stability to the pipeline. Even we did the GWAS to do pre-selection because the data in UK Biobank is SNPs, right? The SNPs, if you look at that level, nothing is stable. Remember I said one thing, we add stability, things became stable. So what we did is that we run the trees in the SNP level, but we interpret it. We interpret at the gene level. So we map the SNPs into genes. So SNP 1, SNP 2 on the same path, we map to gene 1, gene 2. So at the gene level, we saw stability. And that's how we had 5 genes and ranked, include like Titan and CCDC 141, 1GF, some growth factor. And then we did a lot of annotated database. I searched and talked to Ewan's team, and Chad was the cardiology we worked more closely. And then Chad we did the experiments. So, and then we also redesigned experiment because we're also involved looking at how the results. So all of that was, I was holding breath, right? It's like 55% accuracy. One thing I learned is that nothing will pass the significance test at that level, but ranking is still amazingly informative. That's something I saw already in another earlier neuroscience work, my reading with the Jack Galant's lab, like 10 years earlier. There was also the quantitative things didn't make sense, but when we ranked them, to take the top ones, somehow there was information. I don't have a mathematical explanation, but ranking is very stable. It's meaningful. Even none of these genes will pass any significant test.
Interesting. And what was the biological implication of the connection between the genes that you identified, the top ranked ones, and the phenotype? What did you learn? What was a novel biological finding?
I think it's more like to see Titan and CDC 141, right? So Titan is a well-known muscle gene, and then CDC 141 is pretty close to it. And then people thought they always act together. Our gene silencing experiment showed that actually they have their own functions. They don't always work together. They can have opposite effects. And so this is epistasis. But we didn't think, we did more further analysis. We didn't think it's a protein interaction. It's probably some mediation, like indirect interaction. That's what for the follow-up work, we're looking at a lot of rare variants. Seems like random forest, finding a lot of indirect, non-physical. We find some physical one, but it's complementary to this physical type of experiments.
Yeah, interesting.
Yeah, so I think before we did cardiology, we also had a paper called EpiTree. I just want to highlight that. It's using iterandom forest, and go formally we have a PCS p-value, which is a lot more robust than you do testing, use chi-square or something like that. So we come up with a hypothesis, use half of the data. For the test data, we have designed test statistics based on how the interaction epistasis model versus the non-epistasis model in terms of prediction performance. And then we sample the test set, because test set is random split, right? We want to capture that uncertainty. So that was also give us some confidence for human traits, because the first work was on Drosophila, right? It's very clean, simple. But we did red hair first, and then we went to cardiology, which took 5 years. Was a long process, but very rewarding. And we also saw some interaction with growth factor. But experiments, I think, to the further experiments, I think we right now, at least my lab doesn't have access to, and needs more work, right? If people want to follow up for drugs, and there's also something I learned, something called. There's some genes more easily manipulated, other genes are harder to manipulate, so all of that. So I think when we did this work, we didn't take that into account. There's a name I'm escaping me now. Some more you can produce drug for it. So when we go to the next phase, that's something we keep in mind, right? We also maybe want to pre-screen genes based on how accessible for drug development.
Do you have the cardio paper primarily explored like a gene by gene interaction, epistasis, which have traditionally been hard to find. Do you have a hypothesis that there are a lot more of those effects out there for us to find that are really biologically meaningful? And if so, what data or approaches are needed to bring those to light? Because the fact that you spent 5 years to pull this result out just has me thinking about how do we, and this is with a phenotype that you have the MRIs from the UK Biobank, you can look at the, the hypertrophic cardiomyopathy or the hypertrophy in the MRI. What are your thoughts on what we need to discover epistatic gene by gene interactions more broadly?
That's a great question. I think a lot of what I do for the last 10 years now can be called AI for science. And I think a direction there is really integration of a dry and wet lab. We kind of did it in the human level, right? We talked to UN's lab a lot, work very closely, but we're not in the same space, right? And the delay is pretty like many months or something. I think we'll be able to find such FPC interaction. Also beyond, I don't believe it's only pairwise, right? So for the Drosophila, the first case study, we did see very stable third order interactions, right? But I understand the experimental verification at that point was not quite there, also frontier. So I believe actually there are more than second order interactions. But how to get the signal value, signal ratio up, I think it's a combination of better data, right? And then more diverse data maybe, and also at the same time, quick feedback, experimental data, right? So the next phase, if you, in terms of methodology, you have a very quick, which I don't have, like verification, screening out, then I can build that information back into my next recommendation I'll call prioritization line, right? And then that's what I think we'll be able to identify. And also maybe there's a role for deep learning. I don't know, right? It's at least in terms of fitting things, because random forest, if you think about it, it's very greedy, the way we fit it. And one thing could be that deep learning, and then we distill it into some tree form and still use what I believe the tree form interact. Maybe there are other form of interaction too, right? So this is powerful deep learning we haven't tapped into that and distill and to make it interpretable.
Yeah, really interesting.
And some other work we're doing now is building on protein language models, right? That's another thing in the current project we're doing, also with UN's lab, is using protein language models. So that, in some sense, for me, like solving inverse problem in science is kind of like solving a linear equation, right? You need to enough well-postedness. There's lots of ill-postedness. So the high level, how can we bring in well-post information so that the problem becomes solvable in a reliable way? Not to publish a paper, but to really find something. And I want to make a distinction between publishing a paper and doing research. But
and to define inverse problem, that is where it's difficult to generate a solution, but then it's very easy to check, right? Is that the right way to think about it? Easy to check that the solution is correct once you've generated it?
I think I was broader than that. It's more like there's unknown, and then I think linear model people usually understand, right? Or for linear, like least square solving, you need like how many unknowns, you need how many data points, right? Kind of using very high level method to say that you need enough constraints, enough good information so that the problem becomes solvable in a reliable way. You can always do like linear inverse, right? It's just too many of them and it's not really useful.
Yeah.
So in a very, very high level sense, I'm borrowing that metaphor. Yeah.
Can you you mentioned this this framework PCS for veridical data science? The way that I understand this is really about it's less about, is this particular result that I've generated accurate, but it's more about the process as a whole. Is the process by which you've arrived at the result or the model robust to changes? Is it if somebody else picked it up somewhere else and ran it in a way that didn't just precisely do what you did, would they arrive at it? So it's getting at maybe a more fundamental truth, right? Because you're finding something that is very robust to a lot of changes to the process. Is that the right way to think about it, or what would you add?
Yeah, that's a great way to describe it. It's really look at end to end. Take this data science or statistics or machine learning as system science and recognize that it's not just probabilistic modeling. You have data cleaning, you have problem formulation, and humans, even qualified humans, make different judgment calls, right? There's many papers recently, interesting from social science, showing that different analysts make different judgment calls. You have different qualitative conclusions. So I want to make sure that this kind of reasonable human judgment calls are not dictating the final conclusion, because I think it's common sense that you and I work on the data. We have two scientific theory, which we should at least consolidate somehow, right? Instead of you have your truth, I have my truth, and it doesn't make sense. So this is just, before it was hard to do, now with all the software and all of that, it's very easy to run multiple pipelines. And it's related to the multiverse. It's a group of people working on, recognize the same phenomenon. The difference is that we add to that, we also recognize the same phenomenon, but have principles, predictability, computability, stability to guide this work. And maybe you want to combine them. So it's also documentation. We emphasize a lot about documentation. You document, because the context has to come in. You cannot really say what X means without using narratives. You cannot get model checking with reality by just have X and Y's only, right? I think mathematics is a part of it, but it's not enough. You really have to make almost like liberal art type of argument to be persuasive and bring evidence together. So this framework includes all of that, and to make it a case so that humans can believe the conclusions, why things are reproducible. And the book published by MIT Press has a free version, vdsbook.com. VDS means for virtual data science. I should say it's co-authored with my former student, Rebecca Butter. Took us 9 years to write the book. We did many, many versions from teaching and research because the framework was evolving itself. So I had to go back to make it coherent. Yeah.
Very cool. We'll add that in the show notes for people. I want to actually close out here on maybe a more personal level. My understanding is you grew up in China in the middle of the Cultural Revolution, and you had, by probably all estimates, an incredibly challenging childhood, but those sorts of things shape us in fundamental ways. I wonder if you'd be willing to tell that story a little bit and how that has influenced the way that you interact with people and the work that you do today.
Yeah, I think it shaped me fundamentally as a person and also as a researcher. The environment at the time, people was that there was no university for 10 years and there were no books. And lucky now, I grew up with really wonderful family members, a lot of role models. I basically learned by looking at the role model, and many of them were persecuted. And personally, my father died because of the political persecution. So it kind of, since you couldn't really talk to people, I think gave me the ability, and my mom did take me to a lot of adult gatherings I would be watching. And so gave me this habit of analyzing things, analyzing people, and without books, right? Basically, it's all stories you hear from my grandma, mom, and my relatives. And kind of you're forced to think on your own. I also quickly learned that things can be dangerous if you expose yourself too much. So again, became very rich in my own thinking, figuring things out, and felt safe. Until third grade, that a cousin gave me a book on math, and that was like a safe haven for me because there was truth there. There was something so certain. So it really grounded me psychologically to have a place that 2 + 3 = 4, right? It's So and later I think gave me the habit of being very comfortable on my own, which the whole field might be thinking something else. And I also the belief that majority could be wrong. And then just a few of course I do respect people, I hear them, but it's not popularity contest. It's about going through everything through my own brain and be confident. And about that, I feel like I'm in the right direction, even the whole field might not think so. So this conviction of through your own thinking, your own evidence gathering. I was a pure math major, so the way I became applied was really want to see how statistics is useful or not with my own eyes, and not because somebody told me so. I need to see it with my own eyes, and that's kind of. I find it really, really interesting and sometimes more interesting than doing some of the mathematical work, and very rewarding. solving real problems.
Amazing. Well, thank you for sharing that. How does, on the topic of, I guess, change and thinking about things differently, how are you reacting to all the advances in AI and how that might change how we do science? I get the impression that you're thinking about science at a fundamental level. You're working across a lot of different fields at the same time, which tends to have you think less about what's going on in one particular field, but how do we move our understanding forward in general? What are the big things that you expect to change about how we do science as a result? And what are the things that maybe are actually going to be truths we hold on to that don't change very much?
I think that the plus of AI is that a lot of things I think actually we teach in especially elementary classes can be pretty reliable executed by AI, right? But it's also the challenge because we're using these exercises to train the human brain, for them to become a top scientist, right? So that's where the balance has to be discussed and kept. That we don't want to completely out for productivity short term, right? You just outsource all the brain training, the brain thinking. I think that's quite short-sighted. So I had a discussion with some of my students recently. I would say that even for first-year students, some professor might allow students to give harder homeworks, but allow students to use cloud and code and things like that. I would say that I will make half homework like that and half still people need to do on their own.
Yeah, interesting.
And you just have to keep the balance of exercise your own brain, your own neural connections. And the harder question is like, how many such PhD training do we need? And I think we should use it to our benefits, but at the same time, I'm very interested in keeping the bio-intelligence. For me, life, it's only meaningful with humans, and with help, that's fine. But I don't see the future with only artificial intelligence. That's interesting personally to me.
Yeah.
It's just a taste in what life is about. And back that cultural illusion, really one thing is really, there were a lot of horrible people, there were a lot of good people. And that's what drives me to work with other people. And I feel like I'm not excited to only work with artificial intelligence. I try to use it as information retrieval. I don't want personal relationship with intelligence.
Yeah. Do you think that your experience growing up in the Cultural Revolution did you need to learn to read people and understand people at a deeper level?
Yeah, I think because my family was persecuted, I think how you interact with people become very, very important, right?
Yeah.
Therefore, it became a habit. I spent a lot of time studying people later for fun, but just a habit. So when I enter a room, I basically spend quite a bit of energy reading the room.
Yeah, interesting.
And to make sure, in my group, I think it's good I make sure people have a chance to speak. And so it's not just me, it's really knowing where people are and takes energy, but that's just a habit. I cannot stop it for better or for worse. It's for survival right in the beginning.
Yes. I mean, we talked a lot about interpretability in this podcast. So if somebody hasn't had that kind of innate environment, do you have a sense of what someone could do to be more aware or be able to read their environment, read people better? What are the things that you're actually doing to make that a reality?
I think a lot like just engage a lot in human interactions and don't just be the speaker. I tell my client also be the listener, observe. Right, I read novels. A friend of mine really think that Jane Austen's novel have a lot of human study in there, and use all the different forms of human experience to enrich your own understanding of the human around you. But do sample, and do for people more open. Talk about what you think they are and what they think you read them right. Just ask for direct feedback. But you do have to observe. I think a lot of this blasting of science in Twitter, all of that, maybe it's okay, but at the same time, everybody seems to be busier telling what they think. I think we need to also spend at least half time to listen to what other people say. And also when you say something, I think it's better to think a little about it.
Yes.
What's worth sharing before you blast it out?
Yeah. Yeah, that's one of my
Yeah.
One of my specific requests when people use AI, I use it for a lot of things, but when people just paste what it said and don't actually read it and understand it, I think we can outsource that part of your brain. You can outsource the writing part maybe, but the thinking, not so much.
It will really dilute the knowledge base we have, right? I think that's the challenge. How do we use AI to our benefit, but at the same time, not to degrade our own intelligence level too much. It might happen, but hopefully not too much.
Yeah. Interesting. Well, thank you. This was a really great conversation. I really appreciate you taking the time and sharing all your learnings.
Thank you so much. I really enjoyed it too. Yeah, thank you for having me.
Great. And everybody, thank you as always for listening to the podcast, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.